I believe that 30 million of these animalcules together would not take up as much room or be as big as a coarse grain of sand. Antoine von Leeuwenhoek. The image that you're looking at is what is known as a microbial mat. So this particular microbial mat was taken from Puerto Rico, but the location is not important. You can find these throughout the world. And in the top part of the diagram, you'll see kind of the sort of everyday view that you would get without a microscope. But if you look down on the bottom half of this image, you're going to see a cross section through this microbial mat. So each of these numbers over here on the side is indicating for us a different type of microbial, or in other words, bacterial colony. So there's different single-celled organisms that are living at these different depths, and those different organisms are what produce these striation patterns, these different color bandings that you see in the microbial mat cross-section. Now, why does this matter? Well, this matters because there is a certain likelihood, depending on if conditions are just right, for these microbial mats to fossilize. And what I'm going to show you now is a microbial mat that fossilized 3.49 billion years ago. And what you'll notice if you look closely at it is that you see very similar coloration bandings. This is a fossil, so there's no living organisms in this image at this point. They have been replaced by rock, sedimentary rock. But it's preserved the banding pattern. And what's interesting about this is that at 3.49 billion years old, this is among the very oldest evidence that we have of life on Earth. Now, it's certainly possible that there could have been life before 3.49 billion years ago, but if there is, the evidence has either been destroyed or we just haven't found it yet. Now, with the naked eye, we really can't perceive much more than the banding pattern, but since the advent of the modern microscope, we've been able to learn quite a bit more about what these organisms look like at a microscopic level. What we know is that organisms that make up the mats, like the one I just showed you, are made of what we call prokaryotic cells. And prokaryotic cells, there are a few things that we need to know about them for us to continue. So a typical prokaryotic cell is going to have a cell membrane that surrounds it. And the membrane is essentially, what we mean by membrane is that this is a very thin and a very flexible barrier. In addition to the membrane, the typical prokaryotic cell is going to have a region within it. In addition to the membrane, the typical prokaryotic cell is going to have a region within it, which I'm going to call the nucleoid region. And in that nucleoid region, we're going to find DNA. Additionally, prokaryotic cells have some small structures that we find pretty much in all organisms that are known as ribosomes. And ribosomes are essentially just little protein factories. It's one of the most fundamental requirements in order to have a living functional cell. You have to have ribosomes. But in addition, that's pretty much all there is to the prokaryotic cell. There are certainly lots of different chemicals that you're going to find in that cytoplasm. And sometimes you find a few other things. So a lot of bacteria have a little tail-like projection called a flagellum that you'll see. And you could find some other smaller, less important parts. But for the most part, this is our prokaryotic cell. We've got a membrane. We've got DNA. That DNA is in a particular area called the nucleoid region. And we've got some ribosomes. And that is really the foundation of most cell-based life on Earth. So prokaryotic cells make up about 90% of all the cells that you find on Earth even today. And if you go back 3.49 billion years, if you go back to that microbial mat that we were talking about, 3.49 billion years ago, prokaryotic cells would have been 100% of all the life on Earth. Now, one of the limitations of this cell type is that with prokaryotic organisms, they tend to be sort of stuck at being unicellular. So even though there are an enormous number and variety of prokaryotic cells on Earth, they don't really ever tend to form large multicellular organisms. So they're kind of stuck at this level of simplicity. Simple cell, simple organism, one cell per organism. Okay, now there is a second type of cell that we should talk about, and the second type of cell is the eukaryotic cell. Now, the first thing that I should point out is that the eukaryotic cell is not as ancient, it's not as old as the prokaryotic cell. So while prokaryotic cells we saw from microbial mats that they existed about 3.5 billion years ago, the best evidence that we have for the eukaryotic cell seems to put its origin at about 2.7 billion years ago. So it's about 0.8 billion or 800 million years of Earth's history where the prokaryotic cell is already there, and the eukaryotic cell has not yet arrived on the scene. Now, the eukaryotic cell tends to be quite a bit larger than the prokaryotic cell. That's not the most important difference, but it is a difference on the average. One thing that you got to recognize is the eukaryotic cell has all the same parts that the prokaryotic cell has, so we haven't lost anything. There's still a membrane, cell membrane that surrounds the entire thing. It's a thin, flexible barrier. We are still in the eukaryotic cell going to find DNA. It's organized a little bit differently. It's what we call linear DNA, but it's still there. So we still have our DNA. We also are still going to have our ribosomes. So ribosomes are those little protein factories, and you need them for any cell, really. We're still going to find those. And they're spread throughout the cell. I'm just drawing them in one section to keep the diagram neat. So we have all the fundamentals of the prokaryotic cell, but we have some things in addition to that. The most important difference, the most important thing that we gain when we transition to the eukaryotic cell is internal membranes. So what I mean by internal membranes is that while the prokaryotic cell only had the one membrane around the outside of the cell, the eukaryotic cell is going to have additional membranes inside. The most important of these is a membrane that surrounds the DNA. And you may already know what this is called, but just to be sure, the internal membrane that surrounds the DNA is called the nucleus, or the nuclear membrane, and then the whole thing is the nucleus. Now, that's not the only internal membrane. Additionally, eukaryotic cells tend to have this structure called the endoplasmic reticulum. It's usually kind of convoluted and folded, sort of like that. So it's endoplasmic reticulum, which we just say ER for short. There are other structures as well. There's something called a Golgi apparatus and lysosomes and vacuoles, and in plants you find chloroplasts. The names of all of these parts is a little bit beyond our scope right now, and you can certainly look at some diagrams and see some pretty good textbook depictions of a eukaryotic cell. But the important thing is that we have these internal membranes. Whereas a prokaryotic cell has one cytoplasm, and everything the cell does happens in that one cytoplasm, the eukaryotic cell has these parts. It has these subsections. We sometimes refer to these as organelles. But the really important concept here is that they are compartments. Right? We have compartmentalized the eukaryotic cell. We have certain sections, certain regions, certain spaces that are meant for certain functions. So the DNA is stored in the nucleus. The chemical digestion happens in the lysosome. Photosynthesis happens in the chloroplast. We've got these sections of the cell, these compartments, these organelles that are going to devote themselves to specific functions. Okay, that's about it. That's the end of the video. Hopefully at this point you feel confident that you can compare and contrast the traits of prokaryotic and eukaryotic cells.